The Ernst Mayr lecture is one of the clear highlights of the year, not only of the Berlin-Brandenburg Academy of Sciences, but also of the Wissenschaftskolleg, is one of the clear highlights of the year. Dear Barbara Stolberg-Rillinger at the screen, not in our auditory, but dear Dieter Ebert, dear Axel Meyer, dear ladies and gentlemen, the Ernst Mayr lecture is of course one of the clear highlights of this year because you, Hopi Hoekstra, will be speaking. And I would like to welcome you very, very warmly here in the Leibniz Hall of the Berlin-Brandenburg Academy of Sciences and Humanities. The Ernst Mayr lecture is one of the definitive highlights of the Academy's scientific life, not only because it's what founded under the name of our honorary member. The first honorary member, Ernst Mayr, was the first honorary member of the newly constituted Berlin-Brandenburg Academy of Sciences, but also Mayr gave the first lecture in this series here 25 years ago. Personally, it became clear to me sometime before I was elected to the Academy that the Ernst Mayr lecture was a highlight of the academic year when I was a fellow of the Wissenschaftskolleg decades ago in the year 1998-1999. Jared Diamond from Los Angeles spoke here in the Academy building, if I remember correctly, in the Einstein room, not in the Leibniz Hall, which was in the process of renovation. Diamond spoke on the topic of rich and poor. We discussed the thesis of his lecture, which was later published as a small monograph under the title Guns, Germs and Steel, Pulitzer Prize, again and again over breakfast or lunch in Grunewald, Wallotstraße. This was coining the debate of a whole fellows community in Grunewald. And this is not only with my fellows lecture in the year 1980 and 1999, but in a large number of these 2 years lectures in the last 25 years. Also, I have not only been now elected to this academy, and I'm now the president, and therefore have the honor of opening your lecture, dear Hopi Hoekstra. I'm no more a specialist in this subject that has been the subject of these Ernst Mayr lectures for 5 and 20 years than I was with the Diamond in the year 1998. To be more precise, as a historian, not as a biologist, as a philologist and theologian who deals with religions in antiquity, I'm also miles away from the topics of our lecturer this year is dealing with. The last president of the Berlin-Brandenburg Academy, who was really a specialist in the Ernst Mayr lecture, was also the first president of our newly constituted academy. That was the unforgotten Constance biologist Hubert Markel, the last honorary member of our academy. So we are framing our honorary members, Mayr the first and Markel the last. As biologists, Mayr and Markel have never shied away from placing their subject in a broader context, and thus also talking about humanities. Markel likes to be a little bit provocative. I will never forget how he formulated his plea that excellent science today should be formulated in the English language, not in the German or any other non-English language. Without regard for the heavy protesting humanities scholars and their attachment to the German language, and Markel, a very provocative idea, used the Munich Romance scholar Karl Vosseler to justify his argument. He beat the humanities scholars with a humanities scholar. And the first Ernst Mayr lecture by Ernst Mayr on the 14th October 1997 here at the Academy was also dedicated to a topic that transcended the strict boundaries between natural sciences and the humanities. Mayr spoke, some will remember, yes, some will remember, Axel Mayr and perhaps some else will remember. Mayr spoke on the topic, what is actually the philosophy of biology? And not only about anthropology, but also about ethics. It's a quite interesting lecture, and at our e-publications webpage, visible and readable. In view of my predecessor Markel and Ernst Mayr's lecture, I dare to conclude this greeting with a few thoughts. I apologize. Thoughts from a scholar completely outside your field. Historical studies cannot exist without sources, and sources must be edited. Editions are all-day business of my discipline and many others. And for each edition, which is set up in the sense of the classical method to edit texts, founded here in Berlin, in the 19th century by our member Karl Lachmann, that's not so of interest, the manuscripts that bear witness to a text must be hierarchized to identify the best manuscript and to put the readings from the best manuscript in the edition. And these manuscripts should be inserted into a stemma. There is a stemma. And it is the stemma, a family tree of the dependencies of the manuscripts, leading down to the first original, the original text, or an archetypus, which is named Omega. That is important for me, because that stemma, as you probably immediately have realized, is a link between the editorial-based historical sciences and evolutionary biology. There are also lots of stemmata. For such a stemma, as a family dependency connects, if I see it correctly, at least the classical theory of evolution with a classical form of classical philology. Both have these stemmata, I think. Yes, I'm correct? Yes. In some sense, I'm correct. And if at least these two completely different fields are linked by the idea of family trees, dependencies, and stemmata. And in both science, in my own, the editorial sciences, and in yours, and in yours, and in yours, and in yours, and yours, such a stemma is a graphic modeling of family dependence, and that's of a fundamental importance for the establishment of these sciences in the 19th century. So two sciences, the new editorial technique in philology and evolutionary biology, were coined and driven by this idea of it's possible to model graphic dependencies, family tree dependencies in such stemmata forms, and these ideas in two completely independent disciplines revolutionized the biology science and our historical sciences. At a conference on the philological method of editing books at the Wissenschaftskolleg, where else? In the year 2018, the Munich Greek scholar Oliver Primavesi showed that the first corresponding genealogical tree in philology appears in Sweden. Interestingly, there was a kind of manuscript where family trees were shown in a facsimile, and then the two editing Swedish history of law scholars put the family tree of the manuscripts. Now the interesting question, any connections to biology in Sweden? And how are the family dependencies? This is perhaps an interesting question, but not for greeting. This is perhaps for a conference, for a lecture, but that's an attempt to show how in the 19th century, in the history of evolutionary biology and in the humanities, there were comparable ideas, perhaps without any clear-cut contact. Perhaps the same idea at a different place in completely different contexts, but it's an interesting coincidence. And the interesting question is when Mayr, for example, perhaps you and others now as evolutionary biologists are looking and transcending boundaries to the humanities, are we to a certain extent repeating, transforming a situation which based in certain similarities of the 19th century? That's the question, the greeting of the president of Berlin-Brandenburg Academy lays to your feet, in your hands, as a kind of welcome present of the president of the Academy. However, I can reassure you, dear Hopi Hoekstra, ladies and gentlemen, with my little digression on the relationship between philology and evolutionary biology, I did not want to presume to give myself an Ernst Mayr lecture, but only to welcome you all to this year's Ernst Mayr lecture, an anniversary lecture, a specific highlight of Wissenschaftskolleg's year and of our year, a wonderful concluding moment. It's a pleasure to work together with the Wissenschaftskolleg at this point. A greeting again to Barbara Stolberg-Grillinger, but also to those who have been preparing this lecture since the beginning. I name Rüdiger Wehner and Axel Meyer, and also the people responsible now. It's a pleasure and many, many thanks for the efforts you have taken to select this wonderful lecturer of this year. And now I greet you all these I haven't said a very warm greeting before, especially again you. It's a great pleasure to have you here, and wish this anniversary lecture a good cause, and us all good insights into the matter. Thank you very much, and again, a very warm welcome. And now it's my great pleasure to ask Dieter Ebert to introduce really into this Ernst Mayr lecture.
Thank you very much, Professor Markschies. I also want to welcome everybody here to today's Ernst Mayr lecture. As you heard already, it's an anniversary lecture in two aspects: 25 years since the first lecture, and it's the 20th Ernst Mayr lecture. So gives a bit of a feeling being special. Furthermore, we had a break of several years due to Corona, and for me it feels good to be back in activity, be back in a room with many people, and I hope you all feel the same, rather than feeling awkward of having no masks around other people. My name is Dieter Ebert. I'm a zoologist from the University of Basel. I'm a dedicated evolutionary biologist, and I will guide you tonight to this lecture. I will introduce Hopi Hoekstra. Before this, I will say a few more words to Axel Meyer and about this lecture, but I can keep this short because you have heard already various things. So Ernst Mayr was an evolutionary biologist. You can rank him easily among the top 5 of the last century. He was very influential. Born in Germany in 1904, he was already as a child a dedicated ornithologist. Already as a boy, he discovered a duck species which had not been seen in the region where he was living for about 100 years, and he wrote a little publication about this, and this was the beginning of his career. was a red-crested pochard, for those who know duck species. Being a good ornithologist, he had a chance to go to Southeast Asia for a trip where he studied birds there, and he did this so well that he became famous already in his very young age. He went back to Germany. He finished his PhD when he was 21 years old and continued then for some time in a museum, but then moved to America, where he got a position at the American Museum of Natural History in New York. This was 1931. He stayed there for 20 years before he moved to Harvard, and in Harvard he had then the Alexander Agassiz professorship. Remember this name, I will come back to it. Besides being a dedicated ornithologist and having done a lot of work with birds all around the world, mainly in Southeast Asia, he was much more an evolutionary biology on a grand scale. He had conceptual insights which shaped the field more than any other of the people at this time. He was one of the architects of what we call today the modern evolutionary synthesis, which was basically the attempt to bring Darwin's idea of evolution into the next century. What Darwin, of course, didn't know was that there was genetics, that there were genes. So all of this was discovered later. Actually, ironically, Mendel discovered his law almost at the same time as Darwin published his first book, but nobody read it, including Darwin didn't read it. So it took about 100 years until people kind of merged these two fields, and there was a strong debate about this, and Ernst Mayr was one of the important people in this merger between genetics and Darwinian natural history evolution. Two other aspects where Mayr was very important was that he, in contrast to Darwin, who had actually written a book with the title On the Origin of Species, where there's almost nothing in the book on the origin of species. It's all about the process of evolution, but it's not about how species come about. Ernst Mayr was one of the first people who really dug into this and developed ideas and concepts, and he came up with what we nowadays call the biological species concept, which is one way how you can define species, which differed from previous concepts. The second thing for which he will be remembered is he wrote one book which is called The Growth of Biological Thought. And this was published in 1982, and it's kind of a history of biology through the eyes of an evolutionary biologist. And it is important to see this as a leitmotif for this lecture series. This was the idea at the beginning of this lecture series, to understand that biology is constantly changing, we are developing further, and we want to have this lecture series here to see how the development of biology is going hand in hand with the development of our thinking in evolutionary biology, and therefore we invite top speakers in the field to present this. Was already mentioned, Ernst Mayr came here in 1997 to give the opening lectures. He was 93 years old when he came here. This is quite amazing. He died when he was 100 years old in 2005. Let me come now to our distinguished speaker, Hopi Hoekstra. Actually, Danielle Elizabeth Hoekstra, but everybody knows her under her nickname Hopi, so I will stay with this. As you can see from her last name, Hoekstra, she has Dutch roots, but she grew up in California, and she is a Californian girl in all what it has, all the sunshine around it. She's now professor in Harvard, and she is Alexander Agassiz professor. 40 years after Ernst Mayr, she has the same chair Ernst Mayr had many years before. Now, coming from Switzerland, I can't avoid mentioning who Alexander Agassiz was. He was Swiss. So he and his father emigrated from Switzerland to America. Both of them were well known. Well, Alexander not yet, but his father already, well-known ichthyologist. And both of them became professors in Harvard. And so this is where the basis of the Alexander Agassiz professorship goes to. I mentioned already Hopi Hoekstra grew up in California. She also went to do her undergraduates there. She went to the University of Berkeley, University of California in Berkeley. Initially a bit undecided. She had too many interests and couldn't really decide where to go until she heard a lecture about cockroaches, Küchenschaben. And then she fell in love with this, believe it or not. So she went to this professor who is running a lab on studying cockroaches and studied their cockroaches, which was the first hands-on experience doing real biology, and she loved it. She fell so much in love with evolutionary or with biology that she said, that's what I want to do, I want to study animals. But apparently the cockroaches were not the real thing there, because the next choice was then to go to something bigger. She went to the Yellowstone National Park and studied grizzly bears. After 1 year, however, she decided, okay, she goes back to civilization, said goodbye to the grizzly bears, and welcomed mice in her life. This was during her PhD at the University of Washington. From then on, she did various things with mice. After her PhD at the University of Washington, she went for postdoc to Arizona. Then she became assistant professor at the University of California in San Diego, and fairly soon later was hired for Harvard. This was in 2007, and is there ever since. My personal interaction with Hopi goes back about 20 years. We met at conferences and workshops, and I was always fond of her, but I only fall in love with her, in a sense. I only fall in love with her when we were co-teaching a course. I'm running a summer school which takes place in a remote village in the Alps. And for the summer school, we invite every year excellent scientists. And it's quite a demanding job because you are working long hours, and the students are coming from all over the world, and they are the best of their class. And you know, when you have the best of the class, they're also kind of opinionated and having other characteristic features. So a bunch of 30 kids you have to control, but you need to bring to aiming for even higher levels of their education. And so teaching this class with Hopi was a dream team job. It was amazing. You know, after 10 hours, totally stressed out, Hopi is still smiling. We have the biggest crisis there. Students go amok. Hopi solves the problem. And then in the bar afterwards, everybody's smiling, everybody happy. It's just amazing to teach this class with Hopi. And there's this little anecdote that I said to a friend, I wonder what I can do to make sure that Hopi is coming again to teach with me this course here. And apparently Hopi at the same time was thinking, I wonder what I can do that I'm invited again to teach this course. So since then, we do it about every second year or so, and it's wonderful. And please mark your calendar, 2023, no, 24, we said. Good. Okay, but I didn't actually tell you much about what Hopi is actually doing. So you have guessed possibly already that she's an evolutionary biologist, because that's what we are talking all about here. So let me talk you a little bit through what I noted down here on her interest. Actually, I now got mixed up. Before I tell you about what she is doing, I just want to say that not only me recognized that Hopi is great, many people did so. And this resulted in many, many honors she got. And I asked her whether she can send me a CV, and I printed the CV out, and the paper wasn't enough in the printer. It was such a long list. I will only shorten it to a few, just to name among the top things. Hopi is an elected Howard Hughes Medical Institute investigator. She was elected to the National Academy of Science of the United States. She was elected to the American Academy of Arts and Sciences, and this was the last thing, quite recently elected to the American Philosophical Society. And many, many more. and many named lectures and many other honors. So it's really, yes, I'm not alone in recognizing that you're a great Hopi. Okay, now I come what she's doing. So Hopi made great contributions to the field of evolutionary biology. And, or one could actually say even more so to the field of biology. Many of the things she did are classic already now in biology in general, not only for evolutionary biologists. She is particularly interested in focusing on traits which are variable in populations and which might have an adaptive value. What do I mean by this? You see that, for example, there are mice which are light in color, let's say light brown or black in color, and you wonder, why do we have black and brown mice there? Could it be adaptive? So this is really this Darwinian thinking that when we see something, we reflect it in the light of the environment. Could it be an adaptation? So she's asking questions about various traits and what is the value of this trait in the environment. On the other side, she's asking what are the genes underlying these traits? And so there are lots of interesting methods nowadays how we can find the genes underlying these traits. And you know, the genes are the blueprint for coding what the phenotype means. So knowing the gene is very interesting. But not only this, if you know the gene, you may say, well, yeah, okay, you discovered the light switch. You can switch something on and off, and it becomes either black or not black. So what is the value of the gene? Genes have sequences, and the sequences is a long list of nucleotide of letters, 4 letters in the alphabet of the genes, and you can read this. And this can tell you much more than simply this is a gene for being black or being brown as a mouse. There's a history in this gene. And Hopi reads the gene like a history book, taking the gene and asking questions like, how old is the gene? What can we learn about it in terms of, is it a special gene only for this population, or is it a gene which you find also in other species, maybe even in humans? So there's a lot of information you can do there. And you can even go further. You can, from what you see in the gene, you can try to understand what might have been the adaptations in the past. So you can do basically ecology backwards. Millions of years ago, what might have been there? Just looking what our ancestors did, or the ancestors of mice or rats or grizzly bears maybe. So there's a lot of things to do, and you can put all of this together, and you get a comprehensive idea how traits came about and what maintains this trait in the population. Now, Hopi developed, applied, and perfectionated this method to a degree which is beautiful, and you will see in her talk that there are wonderful examples of how she did this. But she's looking for the next challenges. Now, if you think about what we know about genetics from school days, and I guess all of you had somehow a bit of genetics in school days, so think about Mendel, he had the peas, and the peas, they come in 2 colors, green and yellow, or they come in a form which is kind of wrinkly or smooth. All these examples from Mendel, there are always 2 options. There's either one or the other option. But look around, neither here in the room, nor in the forest, wherever you go, do you ever see traits which come in 2 options? Rarely. It's really hard to see such traits. They do exist in the piece of Mendel, for example, and they do exist in a number of other traits, but they're really the exception. The majority is quantitative variation, shades of gray if you want so. Different body sizes, different weights, the eyes, the distance of your eyes, the colors of the hair, whatever trait you're looking at, almost everywhere it's quantitative. Or as many biologists call them, complex traits. And complex traits, as the name implies, has 2 aspects. One is the trait is really complex to study. The other thing is it's an excuse for us not to study it because it's too complex. Too big to ask. not for Hopi. Hopi decided these are important traits, and she started to study them, and with an amazing success. But I will not tell the story of your talk. I rather would like you to tell us what you found out. And so I think I talked enough, and I want to welcome you here on stage, and I will withdraw, and I want to wish you all the best for your talk.
So thank you very much for the warm welcomes. It's such a pleasure to be here. And after 2 years of being postponed, it's even more exciting to be here today. So today what I want to do is tell you a little bit about nature's palette. And in particular, I'm going to start with a historical anecdote to motivate the work I'm going to tell you about. And for most of the talk, I'm going to draw upon research results from our program. But I'll try to bring in examples from lots of different organisms to hopefully have a little bit for everybody. Okay, so trained as an evolutionary biologist, I couldn't resist the urge to start with Darwin, and Dieter, you set this up so nicely. So as we all know, in November 24th, 1853, Darwin wrote his magnus opus on the origin of species. And in staunch Victorian prose, he outlined his great theory of evolution by natural selection. And what's so amazing still to me today is how much he got right, even back in the 1850s. And second, the simplicity and elegance of his argument. This theory rests on 3 main pillars. The idea being if you have a population of individuals and those individuals compete for limited resources, and if there's variation among individuals in their competitive ability, how fit they are, and if at least some of those traits that contribute to that competitive ability are heritable, then populations should be able to evolve via natural selection to novel or changing environments. Now, in his great tome, he outlined evidence, whether that's through careful observation or simple, elegant experiments, lots of evidence for competition among individuals, as well as variation in fitness among individuals within the population. But there was one piece missing to Darwin's theory, and that had to do with heritability. So he surmised that many of these traits were heritable based on the similarity between offspring and their parents. But what he didn't know, as Dieter pointed out, and was unknowable at the time, was the mechanism of inheritance, because of course this was 50 years before Mendel, and still 50 years later until the discovery of DNA. So I want to start today with just a brief anecdote that links Darwin to that second great discovery, that is the 3-dimensional structure of DNA. So here's my attempt to link to history. So what you're looking at here, I don't expect you to read, but just sort of to appreciate, this is Darwin's last publication, published just 2 weeks before he died in 1882, in a little-known journal called Nature, of course, a preeminent journal still today. The title of this article is On the Dispersal of Freshwater Bivalves. And really what it is a natural history observation, a description of a specimen of a beetle, and clamped to the leg of this beetle is a freshwater cockle or clam. So why, you might be wondering, was this published in Nature even back in 1882? Well, it turns out that this solved a big mystery of how these freshwater clams could move around the British Midlands from lake to lake. They would hitchhike like on this beetle and would be dropped off in different habitats. But that's a little bit of an aside. The connection that I want to make today is the story of how Darwin got his hands on this particular very unique natural history specimen. Well, it turns out it was sent to him by a shoemaker, a budding naturalist living in the British Midlands. He sent him the specimen. They had a flurry of correspondence. It turns out this young shoemaker's name was Walter Drawbridge Crick. And for some of you, that may ring a bell, because a century later, that young shoemaker's grandson and his colleague Jim Watson, with importantly key insights from Rosalind Franklin, made that second great discovery, filling in that missing mechanism of how traits are inherited through the three-dimensional structure of DNA, what Jim Watson humbly referred to as the secret of life. And it's really in this DNA code that we find even more evidence for Darwin's great theory, our 3 billion year existence, the shared evolutionary ancestry of all living things, and the topic of what I want to talk to you about today, that is where in the genetic code are those mutations that give rise to differences among individuals within populations, ultimately between populations and between species. So what we're going to focus on today is questions that interested Darwin, like how does all of this variation arise? But thanks to Watson, Crick, and Franklin, we can start to look for some of those answers in the genetic code. So today I want to focus on two main questions. How and why do traits vary in nature? And the framework for this is we're going to talk about the link between genes, traits, and the environment. And as promised, the main trait we're going to focus on today is about color variation. So the first part of this story, I'll tell you about the link between color and the environment, asking the question, why does color vary in natural populations? What's driving that? And then I'll take you from the field into the lab, where we'll ask, how does color vary from a mechanistic perspective? And then once we've made these linkages and we have a much more complete picture, then I think we can do what I consider the most fun part, and that is go back into the wild and ask about how these traits vary within and between populations. So color is one of the most diverse traits in nature. It varies within populations and even between species. It's one of the fundamental ways in which organisms interact with their environment, and we know it's involved in a number of diverse biological processes. So just to give you some examples, color is really important in mate choice, whether that's the brightly colored petals of flowers that are used to attract pollinator and hence allow plants to reproduce, or the animal equivalent is the showy tail of the male peacock who shimmers that tail to attract females. But of course, color isn't just used to attract individuals, it can be used to repel individuals, like this poison frog who is advertising its highly toxic skin. And of course, no good biological story is complete without some cheaters. So there are species that mimic the patterns of toxic individuals, but they themselves are not toxic and don't bear the cost of producing toxin, but benefit from these bright colors. But by and large, one of the primary ways color is used, at least among animals, is in terms of crypsis. In other words, camouflaging individuals from potential predators. But I don't want to leave you with the impression that we always know what all color is for. In fact, there are many canonical examples, like the stripes of zebra, and the utility of which has been discussed and argued and debated for decades, probably centuries, but decades. And recent studies have suggested, quite surprising to many, that one of the primary reasons that zebra stripes may have evolved, or at least a contributor, is that they tend to reduce the likelihood that blood-sucking parasitic flies land on zebras because they prefer to land on solid color objects rather than striped objects. And there are other examples, like that of the color-changing chameleons, which were once thought to use that color-changing ability to camouflage themselves. It's now thought that the color-changing is more of an indicator of physiological condition as opposed to camouflage. So there continue to be exciting updates on how color and patterns are used. There's also diversity in how color is made. We often think of color being made through pigments, but the most striking colors, like that of the blue morpho butterfly or these blue rings on this particular octopus, are not made by pigments, but instead tiny nanostructures that either scatter or reflect light to particular wavelengths, giving rise to these sort of amazing, beautiful colors. But of course, pigments are important, and pigments can be made in several ways, or in this case, acquired from nature through the diet. So for example, these two birds eat plant material. Those plants produce carotenoids, which then are incorporated into the beautiful feathers of either the American goldfinch or the rosy color of this pink flamingo. Another way pigments are produced is they're actually synthesized in the body. So that's epitomized by these two examples. Here, these pterin pigments in this European butterfly come in two forms, either this brilliant white color or this golden color on the tips of its wings. And of course, in other species like mammals, we have melanin, and this North American grizzly bear, and it's the distribution of and intensity of melanins in the hairs that gives this bear its grizzly color. But of all of these types of ways to make color, we know the most about melanin synthesis in mammals. And that's because for over a century, geneticists who are working on laboratory mice often have giant colonies of mice, and every once in a while, a mouse is born with a unique color. And these geneticists can then use genetic mapping approaches to identify the genes that are responsible for this spontaneous mutant. And the result, again, I don't expect you to read or memorize this list, but just to appreciate that we know many genes that in which you have a mutation causes a change in color. And it's not just that we know what the gene name is or its sequence, we often know how those genes interact to produce that variation in color. But if we want to study color variation in a natural setting, we need to turn away from laboratory mice, or even their wild equivalent, house mice. As many of you know, perhaps all too familiar with, house mice that live in our homes actually don't vary that much in color. They're sort of uniformly brown, probably because they're human commensals. So what our lab has done is turn to a close relative of house mice, so we could take advantage of all this amazing knowledge, but look at this color variation in an ecological context. So let me introduce you to the main player of today's story. This is Peromyscus polyanotus, but otherwise known as the old field mouse. And that's because this particular mouse, throughout its range in the southeastern US, so we have Georgia, Alabama, South Carolina, and northern Florida, occupies almost exclusively these overgrown agricultural fields called old fields, which have this dark, loamy soil. And throughout this habitat, the mice look just like this. To many of you, a typical mouse, it has a dark brown coat, a very light-colored belly, and if you could see the tail, it has a stark stripe on the tail. The reason this particular species was of interest to us was because not only do these mice occupy these old field habitat throughout most of the range, they've also colonized these beach habitats and sandbar islands off the Gulf Coast of Florida on the west, as well as the Atlantic coast of Florida in the east. And these mice literally live on the beaches, so I'm going to refer to these as beach mice that recognize where their habitat is. I'm going to start today by talking about one of these subspecies. So these are all one species, but one of these subspecies, number 3 here, the Santa Rosa Island beach mouse. So the habitat, as I mentioned, is very different from the mainland habitat of Florida. This is actually from one of our field sites. We like to travel there in, let's say, February, when in Cambridge, Massachusetts, it's cold and icy and miserable, and we go down to Florida to the beaches. I'm pretty sure this is one of my postdocs taking a little break from their research. As you can see, compared to the mainland mice, this habitat is different, and it's different in 2 main ways. First, the substrate, or the soil, is much lighter in color. I like to think of this as you're almost walking on hills of granulated sugar. It's so white. Second, you can see there's much less vegetative cover. So these mice actually experience relatively high levels of predation, their primary predators being avian and mammalian. And I'll get back to telling you more about the predators in a minute. So it may not be surprising then, as we catch mice on these islands, they look different. And I need to tell you 2 things. One, this is not to scale. I know things are bigger in the US, but not this big. These mice are both about the same size and about the size of a ping pong ball. So they're very small and very cute. But what you can see here is the mice that occur on the beaches are lacking pigment on their cheeks, on their flanks, and if you saw the tail, it's missing that dark stripe as well. The other thing to note is that we know something about the geological history of these sandbar islands, and they're only about 3,000 years old, which suggests that this difference in color and pattern has evolved quite recently. So you must all be thinking, well, this makes perfect sense. If I was a mouse living on these beautiful white sand beaches, having a light coat color would better camouflage me from all of those visually hunting predators. Well, that's a wonderful hypothesis, but as a scientist, we need to be careful not to fall into the trap of Rudyard Kipling, so nicely highlighted in the early 1900s, in making up a just-so story. For example, how the camel got his hump, how the leopard got his spot, how the rhinoceros got his skin, or in other words, how the mouse got his fur color. Instead, what we'd like to do is an experiment. So we want to design an experiment to try to understand why color varies and how the environment plays a role. So if you think about what kind of experiment can you do, here's my dream experiment. What we would do is we'd go out into the field and we'd catch, let's say, 100 beach mice, light in color, 100 mainland mice, dark in color, and then we'd build enclosures on the beaches and enclosures on the mainland, and each of those enclosures we'd put, let's say, put 50 light mice and 50 dark mice. And then we come back sometime later, they're open to predators. We come back, let's say, a month later, and what we would expect is that light mice would survive better on light soil, and there would be more dark mice in the dark soil enclosures, if camouflage was indeed an important component of the story. But you can imagine that's a really hard experiment to do. You'd have to catch lots of mice, you'd have to build enclosures, you'd have to get permits to switch them, and so forth. So I'm going to tell you about what we did first, which I would argue is the next best thing to that experiment, and that is we made mice. We made hundreds of plasticine models of mice, which you can see here, and we painted half of them dark color and half of them light color to mimic the two types of mice. And one benefit of this experiment is those plasticine models only differed in color. They were the same size, they smelled the same, and they acted the same, right? The downside of this experiment, however, was the question of could we fool the predators? Would the predators try to attack these model mice? Well, I'm telling you this story because we could do this. So next, what my postdoc, with the help of a Harvard undergraduate, did is they released equal frequencies of dark and light mice on light soil habitat and dark soil habitat, and then came back the next day and asked, did any of the mice get attacked? And if so, what color were they? Well, here's one of our data points. So here's a dark mouse on light soil, and what you may notice is missing a chunk of its back and part of its left ear. So this was an attack, and it was attacked by an avian predator because you see a nice bill imprint and you don't see any footprints around. So what we could do was simply count up the number of attacks of both types of mice and both types of habitat. So that's summarized here, with these bars indicating the attack rate in each habitat. So if we first focus on the data from the beach habitat, what you can see is if you're a light mouse, the attack rate is much lower than if you're a dark mouse. The same thing is true here when we look at dark habitat. If you match your habitat, your predation level is lower than if you mismatch your habitat. And interestingly, it's just as good to be a light mouse on light habitat as it is to be a dark mouse on dark habitat. So there's selection in both directions. We can actually quantify the strength of selection, and it turns out if you're a mouse and you match your habitat, your chances of survival go up 50%. So this is really strong selection. And then finally, it turns out that about half of the attacks were from avian predators and half from mammalian predators. So both are contributing. So this was the data from this experiment, but remember that dream experiment I told you about at the beginning of making large enclosures and using live mice? Well, we did that experiment too. So this was written up in the Atlantic. Here, what I hope you can see is the steel wall, which represents one of our 8 enclosures that we used to build these giant enclosures, which we then added mice to and did this experiment using live mice. Here's a truck for scale, so you get a sense they're about 1 hectare each. And it turns out, just like our model mice, color matters for survival, even when the mice are wild and alive. So these two experiments together give us some sense of at least one contributing reason why color varies is indeed the role of natural selection, with predators being that selective agent. So next, what I want to do is take you now from the field to the lab and tell you a little bit about the underlying genetics, so asking how color varies. Because this is a diverse audience, I'm going to skip over about a decade worth of intensive lab work and just get to the punchline. So here you're looking at it as a visual representation of the mouse genome, which each of these lines represent different chromosomes with little markers indicating variable spots. But I just want to use this to illustrate that when we try to ask which regions of the genome contribute to differences in color variation, we found 3 regions with differing relative effects on color. Today, I want to tell you about just one of these regions, and that's this one here on the top of chromosome 2. So again, through many years of work, we took this chromosomal region and found a gene within that region, and it's a gene called the melanocortin-1 receptor. And it turns out that I like to refer to this gene as the dream gene, in part because it's very simple in structure. It's about 1,000 DNA nucleotides long. It's conserved among all mammals and most vertebrates. And it's got no sort of funny business, introns, or any complexity. So what we were able to do is sequence this 1,000 base pairs in dark mice, the same sequence in light mice, and ask, are there any differences? It turns out in that 1,000 base pairs, there's one difference. And that one difference changes the protein at one site, this amino acid position 65, whereas all mainland mice that we surveyed have an arginine. All of the Santa Rosa Island beach mice have a cysteine. This got us really excited because there's a perfect correlation between what amino acid you had at this position and your color. But it was still just a statistical correlation. We wanted to do an experiment. So before I tell you about the experiment, I need to tell you a little bit more about the basics of pigmentation biology in mammals. So pigments in mammals are produced from pigment cells, also known as melanocytes. And melanocytes are necessary to produce pigment. So we know that if your pigment cells are absent, or you eventually lose them, or they're repressed, you get hairs that are either white or gray. So for example, these pigment cells derive from the neural crest, so along your backbone, and as an embryo, they migrate around the body. But sometimes that migration is not complete, and they don't come together, and you're left with a little patch of skin, like on the chest of this cat or the blaze on the nose of this horse, in which there are no melanocytes. So if you don't have melanocytes, you can't produce pigment, and that's what gives rise to the tuxedo cat and the blaze on, for example, the horse's nose. There are other cases in which the melanocytes migrate around the body perfectly fine, but they continue to need to be replenished, and they come from a stem cell population. But as we age, those stem cells sort of run out of juice, and the consequence of that is that we have less and less pigment that's made, and ultimately no pigment. We also know that you can repress melanocytes, and you can do that in a spatial-specific way. So in work that our lab published a few years ago, we were able to show that these beautiful white stripes that make these racing stripes down the back of a South American striped mouse is caused by the delayed maturation of these melanocytes, which results in this white pigment, or lack of pigment, down the back. Okay. So that's when pigment goes wrong, pigment cells go wrong, but most of the time these pigment cells produce pigments. And in mammals, we have 2 types of pigments, either eumelanin or pheomelanin. And eumelanin is responsible for the brown to black colors, and pheomelanin for the blonde to red colors. And the intensity of that coloration is determined by the amount of pigment. So you can look at your hair and you can determine whether you have eumelanin or pheomelanin, or had eumelanin or pheomelanin in some cases. And this is about what type of pigments you make. And mammals, relatively speaking, have quite boring hair in the sense that we have one type of pigment that's uniform along the hair. But if we look at other species, even like the mouse, they can switch pigment types. So any one melanocyte can produce either kind of pigment, and they can switch between the two. So a mouse, for example, has a hair that has eumelanin. Then as the hair is growing, you switch to pheomelanin, and then back to eumelanin, which gives this striping pattern. And if you have a dog or a cat at home, you can look at their hairs, because the pigment patterns can get much more interesting. You can change the width of this pheomelanin band, or you can switch between dark pigment to no pigment to light pigment, etc. Okay? Okay. So now back to that gene, the melanocortin-1 receptor. It turns out that it's essentially the molecular switch that tells a cell whether to produce light pigment or dark pigment. So when this gene is activated, you add activator, it turns MC1R on, and it takes information from outside the cell and transmits it inside the cell through a signaling molecule called cyclic AMP. So when MC1R is on, you get high levels of cyclic AMP, and it tells the cell to produce dark pigment. If, however, there's no activator, or if there's a mutation in MC1R, the intracellular levels of cyclic AMP go down, and the cell will produce light pigment. So now that you know something about the role of MC1R, and you go back to think that beach mice have that single amino acid mutation, we could come up with a hypothesis, and that is that mutation in beach mice in this gene might lower the activity of MC1R, leading to the production of light pigment that we see in beach mice relative to their mainland counterparts. And so we could do the experiment, and it turns out this is a relatively easy experiment to do. You can take those two gene sequences that differ by one base pair, you can express those in cells in the lab, and then test the function of that protein. So what we do is we take that protein expressing it in cells, you add more and more activator, and you measure receptor activity simply by measuring the amount of cyclic AMP, that signaling molecule. And what you get is this nice sigmoidal response curve, and this turns out to be the data for the dark beach mice. Now we can repeat that exact same experiment, make one mutation to the sequence, do this, and what we see is that no matter how much activator you add, the receptor function remains low. So this nicely shows that that one nucleotide change causes a change the function of that receptor. The function of that receptor then results in the signaling to produce light pigment, and ultimately that light pigment leads to differences in survival. So we've been able to make that connection between not just a gene, but a mutation in that gene, how it affects color, and ultimately how it affects survival in the wild. Okay, so now that we've made these connections, here's where I think things get really fun, because we have a much more complete picture of how adaptation works, and we can go back out into the field and ask how this happens in the wild. Because this is such a complete story, I am slightly immodestly wanted to show you that this has now been incorporated into many biology and evolution textbooks as an example between genes, traits, and environments, and has even made its way into the popular press. But what I'm even more proud about is that this is being used, at least in the US, as a model for how natural selection may happen in the wild. So folks who have developed this system as a way to simulate natural selection in the wild. So for example, you can have a dark habitat, you can have mice of different colors running around in that habitat, you can add in some predators, and then you can look at how the frequency of the two forms of MC1R changes over time. And as a student, you can go in, you can run the simulation. So these predators are picking off the light mice, which stand out, and the dark allele is going high, but then you can switch the habitat type. Now the dark mice are being eaten, and their dark alleles of the melanocortin receptor are going down in frequency over time. So it's a nice way to illustrate natural selection with a real-life example. Okay. So as I mentioned, we learned something about the genetics underlying this color variation through this mutation in MC1R. But all that I've told you about so far is this one beach mouse population on the Gulf Coast, on the west coast of Florida. So the next question is, we also have these 3 other subspecies of beach mice on the Atlantic coast of Florida. Is the same gene and/or mutation contributing to that color difference? So just by way of reminder, we have these 5 subspecies of beach mice on the Gulf Coast and 3 subspecies on the Atlantic coast. So the first thing we did was go back out into the field again around February again and started to collect mice from the Atlantic coast. So here we are catching mice, and it's one dangling by its tail. We're recording its color variation. So my postdoc is writing down differences in traits and different patches along the body. And then the best part of this is we get to release these mice back into the wild. The mice get a little ear tag so we can follow individuals and look at their survival. And in exchange, they donate either a small snippet of their ear, which you may be able to see here, or a tip of their tail that we use for DNA. Okay, so we did this among all the subspecies, and I'm just going to show you in cartoon form some really important points. So the first thing you might notice is that all the beach mice, relative to the mainland form, are all lighter in color. The second thing I want to tell you is that each of these subspecies within a subspecies, there's very little variation. So I can look at the color of a mouse and tell you what subspecies it is, for example. But the third thing you might notice is the subspecies all look different from each other, at least within the Gulf Coast and within the Atlantic coast. But here was the big surprise. If you were to catch a mouse and show it to me, if you said I caught it on the Gulf Coast, I could tell you what subspecies it is. But if you just told me you caught it in Florida, I would have a 50-50 chance of getting the subspecies right. And that's because these mice turn out to be indistinguishable from each other. Even to a trained beach mouse expert, I can't tell the difference. These mice are indistinguishable, and these mice are indistinguishable based on their color and pattern. So because these mice are separated by about 1,000 kilometers, and we know they've evolved light color independently on these 2 coasts, this raises the question, are they so convergent in their color, these independent evolutionary experiments, because of the same gene or mutation? And this is where working on the dream gene comes back into play, because we could just sequence this gene in those Atlantic coast beach mice and ask, do we find the same arginine to cysteine change? So it turns out we started by sequencing 10 Atlantic coast beach mice. We didn't find it. Then we sequenced 100, didn't find it. And we're close to about 1,000 of these mice. Not one of these beach mice on the Atlantic coast has this cysteine mutation. So that suggests there are independent genetic solutions to this common ecological problem of wanting to be a light mouse on light soil. And in fact, there are no new mutations in this gene that cause the same effect on protein structure and ultimately color. So what that means is that there's completely different genetic solutions. So here I've told you in Gulf Coast beach mice, there's a mutation in MC1R. In the Atlantic coast beach mice that have remarkably similar colors, they don't have this mutation. So I want to end by just mentioning, well, what about other species? So here's where I need a little bit of audience participation. So I'm going to ask you to think about a mammal that is most different in their morphology from a mouse. Any guesses? There's no wrong answers. Giraffe, good guess. Nope. Anybody else? Axel, give me something. Leopard, nope. Whale, good guess. Nope. One more. Think about something big, maybe with a trunk. Okay, so what I was hoping somebody would say, this is the first time I haven't gotten an answer, just for the record, is an elephant. But that's not quite right. I want to end by telling you a story about the woolly mammoth. So many years ago now, as researchers were just starting to sequence DNA from now extinct species. I had a colleague who was working on mammoths, and mammoths are a great species to start to sequence ancient DNA because they're only about 14,000 years extinct, and their DNA/bones are found in permafrost. This is like keeping a vial of DNA in a freezer, best, most stable conditions. So I went to a conference, and I was going to present some of this work on beach mice, and I sat next to my friend who was doing this work on woolly mammoths, and I said, oh, we've got this great result. We found this mutation in this gene. And he said, well, I've been sequencing some genes from mammoths, and I actually sequenced the melanocortin-1 receptor. He had 3 individuals, so that's 6 different alleles. And so we took his sequences and lined them up with our beach mouse sequences and asked, are there any differences? And there was a difference in their mammoths. One individual had 1 allele with a mutation, and it turned out that mutation was an arginine to cysteine change at position 65, which suggests that mammoths, like beach mice, sorry for my ability in Photoshop, but you get the idea, may have been polymorphic in color as well. Now, the experiments that we'd want to do to ask why might they vary, of course, are impossible, but as these 2 papers were co-published together, this didn't stop the press from coming up with all sorts of hypotheses, including blonde mammoths have more fun. So at least they thought maybe sexual selection was involved. But nonetheless, here you have 2 very different mammals that have the same mutation, likely contributing to similar colors. So in one case, within a species, we have different genetic solutions. Now we take 2 arguably very different species, and you have the same change that occurs. And it turns out, as people have been studying this receptor in other species, it turns out it's a hotspot for evolutionary change. It's not necessarily always the same mutation, but there are many other mutations in this receptor that lead to common color polymorphisms, including differences in domestic species like cattle. If you have a Labrador, whether you have a blonde lab or a black lab, a single mutation in the MC1R receptor. With a collaborator, Erica Rosenblum, we had been working on lizards. One of these lizards was caught on the white gypsum sand dunes in New Mexico, the other not. You can probably guess which one that is. This is caused by a mutation in MC1R. And we even know this contributes to hair and skin variation in human populations, with particular alleles being at high frequency in, for example, Irish populations. And back to the ancient DNA studies, now, of course, we have the whole complete Neanderthal genome, which was, of course, that discovery was awarded the Nobel Prize earlier this year. And it turns out when you look at the MC1R receptor in Neanderthals, at least we estimate 6 to 10% of Neanderthals likely also had red hair, noting importantly that this is a different mutation in Neanderthals than in human populations, so it's not just, can't be explained by contamination of that ancient DNA. So together what this suggests is that there's both different genetic solutions and similar genetic solutions, whether it's the same gene or different mutations, that gives rise to color variation that we see across many vertebrate species. So today I've hoped to give you some sense of how we're going about making connections between genes, traits, and the environment, and trying to understand both the ultimate and proximate mechanisms giving rise to that variation. So using approaches like Darwin used, very simple experiments like our models of mice, to experiments that Darwin couldn't even imagine, such as genome sequencing, I think we're getting a much better understanding of both how and why we see that perfection of structure and co-adaptation which most justly excites our admiration, these adaptive traits. So with that, I want to thank you for your attention, and I want to leave you with one other slide. If you had noticed this beautiful array of colors and patterns on the opening slide, in case you were trying to guess what species they were, here are the answers. So thank you very much.
Thank you very much, Hopi, for this wonderful presentation. We have time for questions. I start here in the second row.
I briefly wondered whether you had a personal reason for studying the, and looking at one of your slides, it seemed that you had red hair originally. That's not my question, actually, just a remark. But my question.
But can I answer that? I didn't study pigmentation because I have red hair, but as I started studying pigmentation, I wondered about melanocortin 1 receptor and have sequenced my MC1R, and I indeed do have a, one mutation, which probably gives rise to this color variation.
Actually, I have a question.
Okay.
Actually, I have a personal reason for being interested because I was red-haired, and I have 2 mutations in that.
Oh, interesting. Well, I wonder if we have the same one. Yep.
But actually, I wondered whether in your field experiments with mice, actually, there might be an association also with behavior, actually, and that might even interfere with your interpretation. Would you want to follow up on that? I mean, I could.
I think that's a great question, and in some ways really nicely illustrates why the model mouse experiment kind of controls for that, because the mice, one, didn't behave at all and only differed in color. But you're right that when we have these different variants, we might be interested in color, but they could vary in lots of different ways. In that particular case, too, so here I talked about MC1R because that's the simplest story and the simplest gene. But as I mentioned, we know that there are 3 different genes that contribute to these differences. And one of them is called agouti, and it's actually a gene that physically interacts with MC1R and represses its activity. And so these beach mice have higher levels of agouti. And agouti has been implicated in behavioral differences in actually the same group of mice, too. So I think it's possible. MC1R is one of the reasons I think MC1R is such a hotspot for evolutionary changes is the very end of the pigmentation pathway. So if you mess with MC1R, you probably don't mess up a lot of other systems, like behavior. But agouti, I think, might be the one to look at. I think that's a great question. Good point.
There was a question over there, yeah?
Thanks, Hopi.
Hi, Linda.
Hey. So how many ways can you skin a cat?
That's a great question. And I think that's in some ways it's a hard question to answer. We're very biased in the way we approach this because, for example, with MC1R, our dream gene, you can sequence it in almost every vertebrate because it's so simple in structure. So we have lots of examples of MC1R being hit over and over in birds and lizards and humans and Neanderthals and so forth. If you were to sequence MC1R and you didn't see an association, it's unclear if you would publish that result. So that's one thing. Second, as I mentioned, the second gene, agouti, unlike MC1R, agouti is the not dream gene. It's got lots of little exons. It's got these non-coding exons that are 500,000 base pairs upstream. It's very complex in its regulation. It's more of a typical mammalian gene. So when we first saw agouti, we sort of just pushed that to the side and said, well, we'll study that later. So that doesn't really get to your question directly, but it does suggest that there are lots of biases in answering that. So we'll see lots of examples from, like, once you find something in one, then you can study that in lots of different species. I think that's an interesting question, and only these sort of unbiased approaches, whether it's forward genetic mapping or association studies, will really get at that. But those are also hard studies to get to the actual gene. You can find big regions. And then finally, my final thought on this is that some of these genes that are up in the pathway are probably going to have these, what we call pleiotropic effects. I know you know what that is. And are going to affect lots of things. So you might change color, but you may mess up behavior or development or some other trait. So my guess is that if we could know all of the mutations, that most of them are going to occur in genes that don't mess with other pathways or other traits. And whether we can ever do that experiment to get enough data, I think that's a really interesting hypothesis.
So thank you. I was wondering, you showed that MC1R is necessary. Is it also sufficient? can you do a transgene beach mouse and mutate just that one gene back? Or, because you showed us this list of many, many genes that are involved in pigmentation, so there might be other mutations that also contribute.
Yeah.
So if you just mutate this one back...
we haven't done that. We have done the experiment, or others have started to do these experiments. When you knock out, we know in lab mice, we also know in Parameciscus and preliminary data, if you knock out MC1R, you get light color. We haven't done the reverse experiment because doing transgenics in beach mice has its own set of challenges. But one of the things to mention that goes in that 10 years of work that I glossed over was we took dark mice and light mice and crossed them, made hybrids, crossed them, and then looked across the whole genome which regions are associated. So what that means is there could be small effect mutations that we don't find, but we can pinpoint with pretty good accuracy the regions and then go in and find the genes in those regions. So besides MC1R, agouti, and the third gene is a gene called corin, and I'm just writing this paper up, which acts upstream of agouti and MC1R. Those are probably the three big effect mutations and likely not to be lots of other genes that we're missing. I can also say that we can take the MC1R region from beach mice or from mainland mice and put them, by crossing them, on reciprocal backgrounds, which is like the poor man's way of doing a transgenic, and there we see the effect of MC1R.
I'm not a biologist, I'm an art historian. I have witnessed for a couple of years the discussions in the Wissenschaftskolleg. And it came always to a dead end, so to speak, when deciding about the question of Darwin, if it would be the environmental question, which decides in the end about everything. No doubt about that. I say that, I underline that. But Darwin in his concept had another approach, arguing I dealt much too much time in my life on natural selection. I'm interested in intrinsic madness that has nothing to do with environment. And this is sexual selection. An intrinsic activity that has definitely nothing to do with environmental questions. And that was his point. Our discussions came always to the impossibility to decide this. It went always to the environmental side. And yeah, I put this question, I think it is the 50th time or so. Darwin wanted something else, arguing, no, not my second column. Wallace immediately said, when he proposed that in London, Darwin has become mad. And I ask for Nüsslein Vollhardt, who is trying in these days to strengthen this kind of madness of intrinsic autonomous development and Pram. I witnessed how he wrote his book in the Wissenschaftskolleg, arguing for a new concept of aesthetic evolution.
Yep. So these yep, very complicated topic, thinking about other processes besides natural selection that can produce variation. And I should start too, I think there's even discussions about whether sexual selection is a subset of natural selection or it's really its own thing. I'm very biased and probably not the best person to answer that question, in part because I think so much about natural selection and the connection to the environment. I think we are, however, gaining a growing appreciation for developmental processes that may affect the outcome of traits that maybe we hadn't appreciated so much previously, whether that's sort of developmental noise that produces variation and so forth. But natural selection, of course, really works at the phenotypic level and not how that variation is generated. And so I'm having a hard time answering your question. Maybe we should talk afterwards, sort of to get into the meat of exactly what you're asking. But I think there's a lot of new, often from the molecular world, insights into generating variation and then how that variation is being acted on. Because we can use, as Dieter sort of nicely mentioned, we can sort of read the genetic code, and that's providing new insights that Darwin had no access to into how the process works.
You assumed that the animals or your mice just had those impressions of those colors measured on a physical scale. But do you have many animals we know just perceive colors differently, and some animals perceive more colors than humans do?
Yes.
And my question is, do we know anything about this phenomenon with mice? So is there, do we know how mice perceive colors? Well, we know bees, for instance, they perceive completely differently colors than humans do. Can you just elaborate on this topic?
Yes. So I think your bigger point is exactly right. So everything that I showed you today, right, is seen through the filter of our human eyes and the opsin genes we have and the wavelengths to which they're tuned. So we've always been very biased in the way that as humans, we perceive the world, but many organisms see the world in very different ways. For example, many mammals have cover less wavelengths, so see in more or less black and white. And of course, there's organisms that have even more, a larger spectral range and can see UV colors, for example, including many predators of mice. And they've even shown that sometimes in the urine, there's has UV reflective properties, and aerial hawks can see these urine trails and see where mice are running around and so forth. I have to admit, I don't remember exactly what mice see, but I do know they don't have a bigger spectral range than us. So whether or not they see similar to us or more in the black and white, I should know the answer to this and knew it at one point, but I don't know. But I think in some ways what really matters in that sense is one is lighter and one is darker to some extent. And I know mice can see that difference. But one other question that's sort of related to what you talked about that sort of gets into some of the complexity I skimmed over was here I was talking about light mice and dark mice. But it turns out that the patterns actually matter too. So for example, when we did our model mouse experiment, we put a light beach mouse line down the back of tan color. But we also thought, well, this is so hard to paint. What if we just put totally white mice out there? And it turns out that the totally white mice get eaten more than or attacked more than the mice that have that tan stripe. So I think that what matters maybe to predators more than each other is the camouflage, that light tan stripe on the beach mice camouflages them in some way that sort of surprised us. It's not just about perfectly matching, but that maybe having that dark stripe on the back makes it look less of a mouse. You see a stripe instead of the body of the mouse. So there's lots of complexities both in how they're perceived and then the fine details of their color pattern that I think are really important that I glossed over.
Very interesting. Colors very often have been shown to be epigenetics. And there is a paper out on striped mouse tails in 2006 by François Cousin and co-workers. And they show that this is also a signaling molecule like yours. It's KIT, not the transmembrane receptor. And it's even transmissible. It goes to the sperm. Does your system, it's epigenetic, it's not a mutation. Is it possible that your system doesn't allow this to see it because you're in the wild? Or is it a laboratory artifact? Or is it behind your phenomenon Do you speculate about it?
That's a great question, and I think it's an especially good question because some of the first evidence for epigenetic inheritance came from the studies of mice of different colors, including this gene that I referred to that interacts with MC1R called agouti. So agouti was in some sense the poster child for the genetic basis of epigenetic, at least within a generation, inheritance. That said, so we looked at that gene in particular, but there's nothing in our data that is inconsistent with simple Mendelian inheritance. So, for example, when we bring the mice, same environment, can try to minimize all environmental variation, we see the patterns we expect generation to generation. So that doesn't mean that epigenetics couldn't have an effect, but we don't need to invoke it in this particular system. I think maybe in this particular case that could be true, but I think there is enough evidence to suggest that there are epigenetic traits that happen in wild populations too. But of course we study it most in the lab.
Yeah, Hopi, you showed these, I have a question about the pattern. So you showed the different populations that have different patterns. If somebody hands you the mouse, you know which population it came from. Is there a functional significance to that specific pattern? Is it predator related? Is it drift?
That's a great question. I can tell you what we know. So as I alluded to earlier, having no pigment, being a completely white mouse, doesn't look as good as having some pigment. And we see all the beach mice have some pigment. When we looked at the Gulf Coast mice, they all have different pigments, but some are a little bit darker and are slightly more pigmented. And so what we did was we took soil samples from each of those beachy habitats, and it turns out they're quite variable. They're all much lighter than the mainland, but for example, the population that's the farthest west is near the outflow of the Mississippi, which carries a lot of debris, and so there's a lot of sort of black flecks in the sand and so forth. That happens to be the darkest of the beach mice. And so if you do a correlation, just a correlation, between overall lightness of the mice and sand color, there's a really nice correlation between the two. So that suggests that maybe they're sort of fine-tuned to their particular environment. But we haven't done the experiment, for example, of making model mice with slightly different patterns or releasing mice into big enclosures, and partly because I think the selection is going to be much smaller, and so that experiment becomes more difficult. But that's my hypothesis.
I have a question about the mutation. Does it need to homozygous, or can you see the effect in heterozygotes? And I'm also surprised, since it's a loss-of-function mutation, that it always has to occur in the same place. You would think that there are many other places that will knock out the protein.
Yes, and there are indeed lots of other mutations that affect structure, but both causing it to be constitutively active, and so you get a very dark, in some cases completely black mouse, and others that just turn it down, the function down a little bit, and you get a sort of lighter color mouse, and et cetera. So that's a great question. I believe that it's a recessive mutation. I would say 85% sure it's recessive, and you need 2 copies. The beach mice, because they live on these little beaches, are very depauperate in terms of their genetic diversity, so it would be very easy to imagine that they're almost completely fixed for the derived allele, and so being homozygous. They're partially, they're sort of intermediate on the lighter end of things, so it's not completely recessive. And I think about that in terms of course, a completely recessive mutation is going to be less visible to natural selection than something that has some partial dominance to it. So it's partially dominant, but it's not completely dominant.
But the recessive will have a harder time being incorporated into the population.
Exactly, initially, but then when you have this small population size and they're very inbred, their genomes are much more like human genomes in terms of their genetic diversity, whereas the mainland mice or other populations of these deer mice are like fruit flies. They're like every 100 base pairs there's a new mutation, whereas beach mice are every 10,000 base pairs there's a mutation.
So thank you very much for your wonderful presentation. Getting back to sexual selection.
Yep.
I wonder, do you have any data whether the mates are have a preference for the same pattern? So the males choosing, or females usually choose.
Yep.
Females choose a mate with the same body coloration.
Yeah, that's a wonderful question and something we were really interested in. We haven't done the kind of mate choice experiment to test that in a really rigorous way. But if there was very strong sexual selection that was going on in terms of mate choice, we could see that in their patterns of variability at the DNA level, and we don't see that. So it's a very indirect way of saying at least we know there's not strong sexual selection. That being said, doing the experiment, I think would be the way to, where you actually give them a choice. And that's pretty straightforward to do in mouse studies. We use this Y maze, and you would put a dark mouse on this end of the Y and a light mouse here and release the mouse, and they could go one direction or the other. My guess is that we wouldn't see a difference. And that's just simply because mice are not very visual. They're much more olfactory related, olfactory oriented species. But that's just a guess. And we're often wrong. It's a great question.
Hi, thanks for the great talk. I was wondering, the 5 Gulf mice subspecies and the 3 Atlantic coast subspecies, are they each monophyletic groups?
Yes.
So there was one colonization of the islands that then spread?
So it looks like, so if you make a tree of the subspecies and you include mainland, all the Gulf Coast beach mice are monophyletic. They're all related to each other and pretty closely related to each other, although genetically distinct. So if you're a geneticist, FST is about 0.4 or so they're pretty distinct. Then you have a bunch of mainland mice in the middle, and then separately you have the Atlantic coast beach mice that all clustered together. So we think there's been 2 independent events, one that gave rise to beach mice, and they probably spread across the islands, and one the Atlantic coast, and they spread across the islands. And so that's why we sort of talk about this as 2 independent evolutionary experiments of when you become light, how do you become light? Good question.
Okay, I think we are coming to an end of the discussion. Please join me once more in saying thank you to Hopi Hoekstra for this wonderful presentation. Thank you. And I hand over to the president of the Akademie.
So many thanks for your colorful presentation in the multi-layer sense of the word colorful. That was a real pleasure. I have the impression you already addressed in the answer to Horst Bredekamp, there is a certain need to have other opportunities to continue the questions and answers and discussions, and perhaps there are interests of people. We had a longer time, no opportunity for the Ernst Mayr lecture, so perhaps there is interest of people attending to speak to each other, having seen for the first time. And there is an opportunity. The Akademie and the Wissenschaftskolleg are inviting you cordially to a buffet, and the buffet is behind the curtains. And I hope when ending my words, the curtains will be leveled up, and then you have the opportunity to spend a wonderful evening. Again, many, many thanks, and hope to see you soon for the next Ernst Mayr lecture. Bye-bye.